Breakfast session is going to be chaired by Alice Passoni from Mario Negri Institute, and Alice is also the president of Italian Mass Spectrometry Society. So with this, I'll hand over to Alice, and the MassTech team is here. Have your breakfast and enjoy the talks today.
Okay, thank you, Venkat, for the introduction and also for the invitation. Buongiorno. Just joking, we will talk in English for sure. It's a pleasure for me to chair this session. I'm interested in first person in AP-MALDI application, so the whole session is focused on different applications using AP-MALDI for imaging studies. Let me go to my notes. The first speaker is Professor Gary Patti. He is Michael and Tana Powell Professor at Washington University, and he was awarded in 2024 with a Beeman Medal for his groundbreaking work in metabolomics. And today he'll share his experience in using AP-MALDI applications.
Okay, well, welcome everyone. Thanks for joining us bright and early here. It's Tuesday, so every day gets a little harder to get up at 7, but or whatever time you had to get up to get here. But thanks for joining in this morning. What I'm going to talk about today is providing some background on imaging and how we're taking advantage of the MassTech source. I'm going to first provide just a very high-level overview. I think probably everyone in the audience is familiar with the imaging workflow, but I'll provide just a little bit of a quick context on it. And then I'm going to talk about two other challenges that are associated with imaging, the identification challenge and the quantitation challenge. And then I'll say a few words about how we're trying to address this using the MassTech AP-MALDI source. So we'll start here with just a brief overview to kick things off with what the imaging workflow looks like. Again, I think everybody is pretty familiar with this, so I'll go through it very quickly. But the first thing you do is you take a cross-section of tissue and deposit it on what effectively is a microscope slide. And then you use some kind of desorption beam to raster across that tissue. And you'd like to do it at the highest resolution possible. And you can do it in a couple different ways. You can use a laser beam for MALDI, or you can use a solvent beam for DESI, or I won't talk about it today, but you can also use SIMS. Now, each place that the desorption beam hits generates a mass spectrum, and then you can plot your favorite ion as a function of intensity across the distribution of the tissue, which can generate these images that I'll be showing and you'll be seeing all throughout the morning. Now, recently, one of the things that's been available is the ability to do imaging experiments with either MALDI or with DESI. And often we think about these technologies as which one is better, but our perspective is that they're just really highly complementary. This is an experiment that we did a number of years ago where we compared, this is all of the signals averaged across an entire brain section from a MALDI experiment and from a DESI experiment. And the reason I'm showing you this morning is that to really appreciate that the DESI and the MALDI are allowing us to see different kinds of molecules. You can see, I know the colors are probably hard to see because those lines are so thin, but the molecules that are unique among the DESI and MALDI platform are plentiful. So I don't think that it's either or. I think these are technologies that can be used complementary in terms of in a very complementary way. These are some of the different images that you can generate. So you can go from mouse brain to human kidney tissue, plants. I know something that we like to do, and David Muddyman, who's here as well, likes to do, that's intact whole zebrafish, which is really cool. They're kind of the perfect size because you could put the entire organism on the slide, but they're bigger than C. elegans, which are about 1 millimeter. So those are a little small. But you can also get down to even single cells if you're depositing single cells on a slide, and you can image those as well. There's a couple different things that you can do with the with imaging. I'll just quickly go through a handful of these applications. You can think about the anatomical location if you want to know where a particular molecule is within an animal. This is an example of a phosphatidylserine and that's a zebrafish, and you can see that it localizes to the CNS there. We can think about functional tissue units. This is one of my favorite things to, one of my favorite images actually. This is a spleen from a mouse, and what I'm showing you there is lactate, and you can see that the lactate is high in these proliferating B cells compared to the red pulp surrounding areas, and that's particularly compelling if you think about doing that from bulk tissue. So on the right, that's an experiment that we did on the whole spleen, and you can see that you miss this disparity between the red pulp and the B cells if you're just looking at whole tissue measuring an average, the tissue averaging effect. You can also look at neighborhoods of cells. This is one of my favorite experiments that we did. This is a spinal cord from a mouse with a disease, a model of multiple sclerosis. And we found this is an acylcarnitine that localized to the inflammatory lesions of the animal. So we did immunohistochemistry on adjacent sections of tissue from the animal and found in these regions where we have CD4 positive T cells or we have CD11B positive macrophages and microglia. In those same regions, we had increased acylcarnitines, which is a reflection that we had increased fatty acid oxidation in these regions, which is actually atypical for CNS tissues, which are primarily typically using glucose. And then the last application I'll mention here is you can do drug pharmacokinetics. So this is, I think, another popular way in which I think imaging can be very useful. This is an experiment that we did in the early days of COVID, wherein hydroxychloroquine was relevant, if you remember those days. And we gave this drug to zebrafish, and you can see on the left, those animals were treated, and on the right were control animals. And then you can see where the drug goes within the animal, and you can measure not only the drug, but you can measure its transformation products throughout the animal to see exactly where things go. So I think another application by which you can use the imaging technology. So that's kind of just an overview of some of the different things you can do with imaging. But what I'd like to now do is focus on a couple areas where there are challenges that we face as a field. And the first challenge I'd like to talk about is the identification challenge and how we go about trying to figure out what it is that we're actually looking at. Trying to identify small molecules has a very storied history in metabolomics and lipidomics. It's something that has received a lot of focus and attention. And in 2007, from this seminal paper that was put forward by the Metabolomic Standards Initiative, this paper was provided as some guidance for how we go about making, or at least communicating confidence when we make identifications. And so this is still used, or some variant of this is still used in metabolomics today. Okay, and what you see here is level 4 is what's termed an unknown structure. That means that we have very low confidence, and that identification is made by accurate mass only. If you get MSMS data and you can match a fragment or two, or you get some sort of characteristic retention time, then you can get into a possible structure, which would be communicated as level 3. If you're able to get MSMS data, retention time data, or CCS data, collisional cross-section data, and you can match that to reference data from a database that are outside of your lab, then you can get to level 2. But if you want to get to the absolute highest level of confidence, then what you have to do is from your research sample, you have to collect accurate mass, MSMS data, and retention time or collisional cross-section data from ion mobility, and match that to a standard that's in-house, a standard that you run in your own lab, an authentic compound. Okay? And for metabolomics and lipidomics, really we've gotten to a point in the field that's matured over the last couple of decades. We've got to a point that if you're going to be publishing something, you really want to try to get to level 1. Okay, or at least level 2. However, with imaging, we haven't reached that state yet. And when you look at a lot of the literature that's out there in imaging, you see a lot of identifications that are made simply on the basis of accurate mass alone. And so I'd like to just take a moment and review some of the challenges with that and some of the perils that we face. So for example, this is a mouse image of an M/Z of 259.022. And you can see that if you were to acquire MSMS data on this pixel over here, which is the bottom left, you get MSMS data from your sample that matches beautifully to MSMS data of a standard. However, if you just got MSMS data in that pixel, and you said, okay, well, it's a perfect match, therefore 259 is always the same compound across the whole tissue, you could be in danger because you see when you get MSMS data on the top right over here, more in the corpus callosum area, you see that the sample no longer matches the standard. So that's because you've got some other different structural isomer now that is predominantly contributing to the 259 signal. And so now you've got a different compound. So I put earlier the standard MSI standards initiative for how we communicate confidence with LC-MS-based metabolomics, but I think what's really needed is ways to communicate confidence with imaging. While I think it's a great start to be able to use accurate masses, you want to be very careful about how reliable those identifications are. And I think one of the reasons why we've done that is it's been challenging to be able to get, we don't have, the software isn't available to do, the kinds of triggered DDA kinds of experiments that we would want to do to be able to get MSMS across different pixels and that sort of more complicated experiment. So one way in which we can try to address some of these issues, and this is an experiment that we've really been excited about performing, is coupling imaging with the triple quadrupole mass spectrometer. And you may be familiar with the triple quadrupole, but I'll just briefly highlight the experimental design here and talk about its potential advantages in the context of doing imaging. So you've got all these ions that are coming in the instrument here, and you use quadrupole 1 to select the precursor that you're interested in, and that allows you to get, for this example, let's say that the precursor, you have the two structural isomers, the two sugar phosphates that I showed you on the prior slide, represented here as red and blue. Then you fragment both of them, but then you use the third quadrupole to filter a fragment that's unique to either the red or the blue ion, in this case, whatever the precursor is that you're interested in, and then only that fragment, which now has a unique M/Z and a unique fragment, hits the detector. Okay, so you can get the specificity. And we've been doing these experiments with the MassTech source. This is an image from our lab where we have the MassTech source hooked up to a 6595 from Agilent. We've been getting some really beautiful images, and I'll show you some of that. I'm really pleased with how these experiments are going. This is, of course, the AP-MALDI, but I want to make a couple points here that we're doing these experiments with triple quads, but just before the session started, we were talking about how we can also take this source off and put it on our QTOFs. So one of the really cool things I think about the MassTech source is its flexibility. You're not buying $1 million instrument and then stuck with it doing imaging in just that mode. You can take it and put it on, if you're fortunate to have multiple mass spectrometers and you want to do an ion mobility experiment, you can put it on your QTOF with an ion mobility. If you want to do a triple quad, you can put it on your triple quad instrument. And you may wonder how long it takes, and this was actually what was most surprising to me, was how quick, because we have played around with these sources, prior iterations from other third parties, and it's always been kind of very complicated to get it up and running and to tune, sometimes taking a day or two or even longer to calibrate. But this one is impressively efficient. In fact, when the instrument was first delivered to our lab, Constantine, who I think is here somewhere, by the time I could even get down to see him, to shake his hand and say hello, it was already installed. So I thought, well, it'll take him a couple hours to get everything out of the box. And I got down there and it was like already up and running. Is how quickly this was installed. So really been very pleased with that. You can get spatial micron, spatial resolution up to 25 microns. And there's other software options out there to support this. And Professor David Moneyman, who's one of the pioneers in imaging, has actually developed a package as well, MSI Reader. And so I encourage everyone to speak with him if you're interested in that software package. So there's options out there to support it. Now, when we think about how we do these experiments in the context of the triple quad, this is a full MSMS pattern for adenine. So this is the full scan. And if you want to do the triple quad experiment, which you could imagine doing, the 134 is the precursor there, and you see there's a couple different fragments. You can do multiple transitions. You can do the 134 to 92 transition, the 134 to 107 transition, and you can see that you get concordant images, which is great, right? Which means that those fragments are both coming from adenine as we would expect. But you can do the same types of things that we do when you do LCMS-based triple quad. We have a qualifier and a quantifier on, you know, the same sort of business. But there's some real appeal for doing this, at least from our perspective, for imaging, because you get better identification confidence. You're collecting MSMS across the entire tissue, where you try to get to those higher levels of confidence. It also can avoid interferences. We'll talk about that here in a couple of minutes. And the triple quad, you know, is known for its sensitivity, its duty cycle, and you take advantage of that sensitivity, that quantitative reproducibility. So you're really taking advantage of those properties of the instrument. So I'd like to just show you in the last couple minutes here of this section an application of citrate and isocitrate. Obviously, these are structural isomers of one another, and you can see that they have different MSMS patterns. So that's what I'm showing you there is the MSMS pattern for citrate and the MSMS pattern for isocitrate. These are notoriously difficult molecules to separate if you don't have LC. Now, if you have LC, of course, you can get separation through chromatography, but if you're not doing any separation, now you've got two structural isomers. But as you can see there, the MSMS patterns do have some differences, but they also have some similarities. One fragment that's shared between citrate and isocitrate is 111. So if you do a transition from 191 to 111, you're going to measure citrate and isocitrate. However, if you're focusing just on the transition of 191 to 87, then you're selectively going to measure citrate and you're not going to measure isocitrate. And conversely, if you look at the transition of 191 to 73, you selectively measure isocitrate and not citrate. Okay, just as a proof of concept to show you how well this works, this is a slide in which we've deposited isocitrate standard on the x-axis and citrate is on the y-axis. And you can see by looking at the 87 transition, we uniquely measure citrate. And by looking at the 73 transition, we're uniquely measuring isocitrate. Okay, so very appealing, allows us to try to resolve these structural isomers in the context of imaging across the entire tissue, not worrying about are we biasing ourselves in that one pixel, but across the whole systemic tissue. Now, the last thing I'd like to talk about is another challenge that's associated with imaging, and that is the quantitation. And to kick this discussion off, I'm going to reference one of my favorite papers of all times from Roy Goodacre's lab in 2007. And this is a fantastic paper. What he did is he took a complex sample and he added standards to it at different concentrations. Those standards did not include histidine or malate. There was no histidine or malate added to these complex samples. Okay, obviously histidine and malate were present in the complex samples, but additional histidine and malate was not spiked in. Something like 30 other compounds were spiked in at different concentrations. And you see, because these other molecules were spiked in at different concentrations and due to their matrix effects, it actually appears that the concentration of histidine is changing, and it appears that the concentration of malate is changing. Okay, again, those aren't changing, but this is due to the matrix effects. So one of the things that we like to do with imaging, this is a fantastic zebrafish image. One of the things we like to do with imaging is say, oh, look, you know, this is our favorite metabolite, and this metabolite is really low in the eye, and it's much higher over there in the liver, and it's really high in the spinal cord, you know, relative. Try to make these comparisons. But what I encourage you to recognize is that each region of, particularly if you're looking at something like, you know, a spinal cord and an eye, or a liver and an eye, those are very different matrices. So how well a particular molecule is going to ionize across the tissues is going to be variable, and you have to take into account whether or not the differences you're seeing are actually due to concentration differences or whether or not they're due to more complex matrix effects. And just to give you an example of the kinds of troubles you can get into, I'll show you this image. So what I'm showing you on the left-hand slide here is a brain. This is a mouse brain. We injected GL261 cancer cells, and it forms a glioma tumor. And the cells are labeled with RFP. So if you do microscopy, you can easily on the left see the non-mass spectrometry images where the tumors are red. And then we use MALDI and DESI to look at the abundance of a fatty acid palmitate. And you can see that by MALDI, it appears that palmitate concentration is lower in the tumor. But when we take the exact same tissues or adjacent cross-sections of the exact same tissues and analyze them by DESI, it actually appears that the fatty acid is higher in the tumor. Now, the fatty acid can't both be higher and lower in the tumor. Only one of those answers is correct. Which of the answers is correct, I don't know. We're not going to get into that today. But this is an example of the kinds of, because the matrix effects are different with MALDI and DESI, you actually get opposite answers here. Okay, so how do we get around this? And this will be the last topic. I realize I'm running up against time here, but how do we get around this? And the one way in which we can get around this is by applying stable isotopes. So we'll call this the isotope tracing alternative. And the nice thing about stable isotopes is that you can learn a lot from them. So in this particular example, what we're doing is we're giving animals C13 labeled glucose, so the 6 carbons in glucose that are normally C12 are replaced with C13, and then you can track where those carbons go in downstream molecules, such as N-acetyl aspartate, which has 6 carbons, and you can say how much of NAA has no labels, how much has 1 C13, which is M1, how much has 2 C13, which is M2, etc. And that's the LCMS result there, and you can see that the AP-MALDI result looks exactly the same. And why is that? Well, the isotopes aren't subject to ionization effects because we're taking the ratio of the isotopologues. Okay, so you don't, because isotopologues have the same ionization efficiencies, you don't have to worry about matrix effects interfering with this particular pattern. And like I said, it's very appealing to think about how we can use isotopes, because not only can you assess abundances by adding up all the isotopologues, but you can actually track the kinetics. You know, how quickly something is propagating through a pathway, where it's going, is it going to the left, is it going to the right, which pathways it's entering, and how quickly it gets there. And that can tell you a lot of information that you can't get from abundances. This is a paper that we published almost 10 years ago now. And what I'm showing you in the white are cells that were quiescent, and in the, in the white, cells that were quiescent, and the red, cells that are rapidly proliferating. And we know the cells that are rapidly proliferating have higher rates of glycolysis. But when you just measure glycolytic intermediates, you see that the abundance doesn't change, even though we know that that pathway is up. And you have to use isotopes to tease that out, because the isotopes get transformed faster. The abundances don't change, but the turnover rate of the pools is changing faster. And so by using isotopes, we can observe that. Now, the most complicated thing when you use isotopes, of course, is you have to administer the isotopes. And for some, that might be a showstopper, but it really, once you start doing these experiments, they really are straightforward. We use the same syringe pump that we use to infuse standards into our mass spectrometer, but we infuse isotopes into the jugular vein of the animal through a cannula of a mouse here. Zebrafish are really easy. You just add the isotope right to the water. It's like fish food. You just put it on top of the water and they take it up. And we can also do these things in patients using IVs. Okay, and this is just the last couple slides I have here. So this is an example where we've given animals C13 glucose, and you can imagine trying to plot how many labels get into palmitate. So this is the same kind of thing I showed you before. How much palmitate has no labels, 2 labels, 4 labels, and you can make these so-called isotopolog plots. And this is the way we would do it with LCMS. But what we can do with the imaging approaches is we can extend this and we can make isotopolog plots for every pixel. Okay, so this is 4 replicates, 1, 2, 3, 4 different replicates across the top, and then each line there represents all of the isotopologs for those replicates. So you get isotopolog plots like I've shown you on the left that you would get from LCMS, but now you get an isotopolog plot for each pixel of the image. Okay, and that allows us to do these kind of dynamic measurements across the tissues. Now, the last point that I'll make here is that the most complicated aspect of doing this is that you get interferences, because now you have to be able to resolve each of the different isotopologs. And whenever you do these experiments, it's absolutely critical that you do an unlabeled control. So on the bottom, what I'm showing you there is an animal that's been treated with C13 glucose, and we're plotting the isotopologs of NAA. And on the top, that's an unlabeled brain. And the fact that you still see an M5, so if you look over there, here you can see that there's an M5. There should not be an M5 because this is an unlabeled brain. There's no reason that there should be an NAA that has 5 C13. So this is an interference, and that means that we can't trust the M5 from the labeled samples. So you really have to run these unlabeled controls. and figure out where your interferences are. Now, there's a couple ways you can try to minimize these interferences. One is to throw your AP-MALDI on an instrument that has ion mobility. And we've done that, and the ion mobility can help resolve some of those interferences because they have different collisional, different drift times. But you can also move to instruments that have higher resolving power. And again, the beauty of the MassTech source is that you can say, okay, I'm going to move this over to an instrument like an Orbitrap where the resolving power is higher and it helps clean some of this up. And so I'll conclude here by showing you these fluxes. What we have on the left is that same glioma brain. You can see the tumor that's red, but now we can actually measure the flux of fatty acid synthesis. So we're not measuring the abundance of the fatty acid, but that's how fast that fatty acid is being synthesized and how fast it's being elongated on the right. And so you can see that those fluxes are elevated in the tumor relative to the other tissues, the healthy surrounding tissue. So with that, I'll go ahead and wrap up, and I'll thank the people in the lab who are responsible for this work, in particular Mickey, Ethan, Vika, and Kevin, and of course MassTech for their support. And thank you again for coming, and I'll turn it over to the moderator.